I've had such a great opportunity to connect and spend time with a lot of our scientists, our researchers, our medical professionals, and they're all just incredibly amazing people. Everybody's different. I've noticed two major notable consistencies across our community's researchers. Number one, they have incredible ability that you've seen today to share complex scientific information with some of us, like myself, non-research types, in a way that doesn't dilute the meaningfulness, but never feels like they're talking down to us. And they also, number two, they have a tremendous devotion to the work that they're doing, and you can see and hear it immediately when you talk to any of them. And, mentioning that because our next speaker up is Dr. Yetta Lee, and she is the embodiment of both of these characteristics. And if you haven't had the chance yet to speak with her, I really encourage you to do so. She's one of the most kind and intelligent and insightful people I've ever met, and I think our community is very fortunate to have her.
Okay, I am short. Let me not break this.
Pull the whole arm. Pull the whole arm.
Pull the whole arm. Okay. Good enough. Good enough. Oh, it's that thing. Yes, very intelligent here, obviously. Okay. Thank you all very much. It's a pleasure to be here. You know what? Okay, this is generally not how my clinic goes. Maybe I'll just hold the microphone, okay? It's a pleasure to be here, and it's an honor to be here. Thank you so much for having me. So today I'd like to share with you this project that I've been working on for about a year, actually, and I have an update for you. A lot of you were at the Sisters Hope Foundation meeting last year, and I sort of talked about this a little bit, and at that time I just started this project. So I wanted to talk a little bit more about this today. But also, as I have seen a lot of patients in clinic, I've realized that we geneticists like to use a lot of jargon. So we talk about mutations and variants and C dot and P dot and DNA and protein and blah, 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 blah. And I realized that I need to actually just explain it in a way that is not in jargon. And everybody knows me. I apologize if I use jargon in my clinic. So let me start out with that, okay? I am on the board of GLIA Connect and the chair of the Medical Scientific Advisory Board, both unpaid. Okay, so a little bit about this. I'm preaching to the choir, but there are always new faces, so I'm going to give a very brief introduction. So CSF1R related disorders, this is a genetic brain disease that causes destruction of myelin in the brain. Myelin is the insulation of the nerves and allows the neurons to talk to each other in the brain, and basically allows us to do and think and be ourselves. So when you have a mutation in CSF1R, it can affect your thinking, affect your personality, affects your mental health, talking, walking, moving, urinary bowel movements, basically everything about our lives, as many of you have experienced. This disease can result from one mutation or two mutations, or sometimes more, mostly one or two, in the CSF1R gene. So I wanted to talk a little bit about the word mutation because this is a word we use a lot. And I really hope the colors work here. Okay, good. Okay, so let's start with DNA. This is what molecular biologists call the central dogma of biology, how DNA makes up everything in our body, okay? So I'm gonna start with DNA. DNA is made out of 4 letters, A, C, T, and G. They come together and you have billions of letters in each cell. And we call, so the letters can be grouped together into genes, and CSF1R is a gene. And the way I like to think about this is like a big recipe book that was passed down through the generations, so it is very precious, and every gene is a recipe, okay? And so you have this big recipe book in a vault, and it's too precious to actually take out to look at the recipes when you want to make something nice. So what happens is that you make a copy of it. And the process of copying that out is called transcription, and that's how you make RNA, which is the copy of a recipe, okay? So then you can take it out, and then from that RNA, that's the instruction to make the protein that actually does the work in the cell, okay? And so how does the cell do this? How does it read RNA and turn it into protein? So RNA is read in chunks of 3 letters. Each 3-letter group stands for an amino acid, and amino acids are kind of like the ingredients you use to make a dish, okay? So when you put all the ingredients together in order according to what the RNA says, then you get the protein, okay? And here the protein is also called CSF1R, CSF1 receptor. When things go wrong in the DNA itself, okay, in that big recipe book, you can get mutations, and because of this entire process, you get changes in the amino acids. And depending on where the mutations are, you can get different changes in the amino acids, and these are called mutations. More recently in the field, the word mutation has now been replaced by the word variant for multiple different reasons, but for all intents and purposes of this talk, mutation and variant are the same thing, okay? Okay, so how does this relate to the genetic report? Because I know all of our patients get genetic testing, everybody gets a report, and what are the components of the report and how do you read it? So in every genetic report, it will tell you which gene has the mutation. So that's CSF1R. That, again, is the DNA, okay? And then it will tell you what mutation it is. So this is where the C dot and the P dot come in. The C dot is the actual DNA mutation, so in that big book in the vault with the recipe. And then the P dot is the protein mutation, so how that actually changes the dish or the protein that is being made. The genetic report will also have something called zygosity, which just basically means, do you have 1 copy of that mutation or do you have 2 copies of that mutation? And there are many different words used to say this, okay? So I put them on here. Heterozygous or monoallelic means you have 1 copy. Homozygous or biallelic means you have 2 copies, okay? And then it'll give you a classification of the mutation. So there are many different types of classifications. I put variant of uncertain significance here because this is actually one of the very common classifications, and this is kind of a problem in the field because you don't really know what to do with that. So transitioning into this, so in addition to a variant of uncertain significance of VUS, the classification can also be benign or likely benign, and pathogenic or likely pathogenic. And for all intents and purposes, in clinical work, benign and likely benign are treated as the same, and pathogenic and likely pathogenic are treated as the same. But if you talk to geneticists, these actually have very specific definitions that have been mathematically calculated as a probability. So when your mutation comes up and it's likely pathogenic, that means there's over 90% certainty that this mutation causes disease. And if it's pathogenic, there's over 99% certainty. Okay? Now, from a clinical perspective, that to me, for clinicians, is the same. Okay? So that means it causes disease. And I should say, the word pathogenic means it causes disease. So, after explaining all of that, the question that I get most in clinic in various forms is, what does my mutation mean for me? Because most people have different mutations. So in order to answer that question, this was a question that has really plagued me a lot because it's sometimes very hard to answer this question because of the variant of uncertain significance. And so this is how the idea for the International CSF1R Variant Database got started. I wanted to see, just by looking through literature at first, how many case reports and case series were out there for patients with CSF1R related disorder, how many mutations I could find and compile into a list and put all the information together into one database so that all the clinicians can then go and easily refer to it to look to see whenever a patient comes to see us in clinic. Now that has since expanded. So I did a literature search, identified 397 variants from there, and then I also looked in the databases. So these are databases that genetic counselors use to interpret the mutations to see whether it's pathogenic or benign or a variant of a certain significance. And then I also collaborated with many international collaborators, so many thanks to them, who also submitted patient cases, all anonymously of course, and gave me variants to include in the database to make the database, one, international, which is really important, but two, as complete as possible. So far, we've identified 434 different variants or mutations in CSF1R. This piece of data is really important. So ClinVar is one of the most common databases, public databases out there that genetic counselors and clinicians use to interpret variants to see, is this mutation disease-causing or not? And what was shocking to me was that only 41% of the 434 variants were even on ClinVar. 59% of the variants were not even on ClinVar, so you couldn't even find them on ClinVar. Which brings me to the point that the ICVD, so this is expert curation of mutations, it fills a major gap in the existing databases. So this slide shows data that I presented last year at Sisters Hope Foundation for the number of publications and patients and mutations that I had identified. So I just wanted to give an update. It is now 2026. It's almost been exactly 1 year since I last presented these data. We now have 335 publications. There are 19 collaborators from around the world representing, the mutations represent 45 different countries. We have 434 new unique mutations, 1,492 patients with positive genetic tests confirming CSF1R mutations. We have 778 patients with confirmed symptomatic ALSP, or one copy of the CSF1R mutation causing disease. There are now 36 bandose patients, meaning patients with two mutations in CSF1R, or at least mutations on both copies of the gene. We have 153 asymptomatic carriers, 160 patients with a CSF1R mutation who were diagnosed with other diagnoses. We have 365 patients with positive genetic tests whose symptoms were not reported. So this is the point I really want to make, okay? And I think this is true for a lot of rare diseases. You are not alone. I think this disease is definitely not as rare as people make it out to be, just given how many patients have already been identified in the last year through literature search and collaborators. And this is also kind of makes the same point. Now, CSF1R related disorder has been found on all 6 continents, all 6 inhabited continents. Okay, and I suspect the numbers will only go up as, 1, awareness increases, and 2, we do more genetic testing for adults. So we have 434 mutations. What do we do with them? So for clinical care, it's really important to classify them and determine whether they are disease-causing, okay? Because that's going to influence clinical trials. That's going to influence how we clinicians counsel patients and make sure that we give you accurate information. So these classifications are from the ACMG AMP criteria. That's the American College of Medical Genetics. Okay, the criteria were published in 2015. Okay, There's a couple of ways to look at the classification. One way is through ClinVar, and that's, again, the most commonly used database that all clinicians and genetic counselors use. Okay, so ClinVar will tell you whether the mutation is pathogenic or likely pathogenic. Sometimes it'll have two categorizations, and that's because these are from laboratories that submit the mutations, and then each laboratory has a geneticist who looks at the mutation and determines whether or not they fit these criteria that were published in 2015 and whether the mutation is pathogenic or not. Okay. Another way to look is through these in silico predictors. What that means is that it's a computer program that looks at the gene and does a whole bunch of algorithms that, frankly, I don't understand, but basically looks at all the criteria published here and classifies each criteria and then makes a prediction as to whether this mutation is pathogenic or not. Okay, so these are two existing methods. Now, what I have done is taken all 28 criteria published by this paper, okay, and modified them specifically for the CSF1R gene. So this has been done for many different genes. It has not been done for CSF1R yet, but this is really important because every gene is different and every gene needs its gene-specific criteria. So looking at these manual classifications, so I call those the manual classifications, okay? I apologize, the colors definitely didn't quite come out right. Okay, so let's look at this. This is easier to read. Okay, so there are about 10% of variants, mutations that were benign or likely benign. There were 39.2% of mutations that were VUSs, and 50.9% that were pathogenic or likely pathogenic. So then the next question becomes, okay, so these are manual classifications. Now, I mean, did they clarify any of the VUSs compared to what ClinVar and Franklin, called? Is this better in terms of interpreting what each mutation does and whether each mutation causes disease? So, VUS clarification. So there were 240 VUSs from ClinVar and Franklin, and there are now 170 VUSs, okay, which means we clarified about a third of the VUSs, about 33% of the VUSs. So it's an improvement, but there's still a lot of work to do. And I really want to emphasize here, your story could be someone else's answer. So one of the ways that is really important for upgrading these VUSs is to have more than one patient or more than one family who share the same mutation and have a history of disease. And that goes a really long way in upgrading these VUSs to likely pathogenic or pathogenic, which is why it is so critical that every single patient matters, and we really need data from every single patient. Okay, so looking at manual reclassifications in a little bit more detail. So the last slide was on the VUSs. Here we are looking at the change in classification, comparing the manual classifications to the ClinVar classifications and the computer-generated classifications. And so the most important part on this slide, what I want you to focus on is here, okay? So remember, there were 59% of the variants were not on ClinVar, so you couldn't find them on ClinVar. And unsurprisingly, the majority of those were the ones that got upgraded to pathogenic or likely pathogenic, okay? So what that means is that the least known mutations, the mutations with the least data and the least publicity, those are the ones that benefit most from expert curation. Okay, I want to talk a little bit about disease penetrance and what that means. Penetrance is defined as the percentage of patients who develop symptoms divided by the total number of patients who have a mutation in CSF1R. Okay, and it's less than 100%, but we don't have a good calculation for exactly what that is. So in other words, it answers the question, if I have a mutation in CSF1R, am I going to develop symptoms at some point in the future? What is my chance of developing symptoms at some point in the future? And so I want to talk a little bit about exactly what penetrance is, because it's a little bit more complicated than that, okay? So let's say you have 10 people here, 5 of them develop symptoms. So then the penetrance here would be 5 out of 10 or 50%. And let's say that's at a specific age, because over the lifetime, the penetrance changes, because as people get older, more and more people are going to develop symptoms. So for example, at a very young age, and I want to be totally clear, these numbers are not for CSF1R. I just made them up to explain what penetrance is over time, okay? So at a very young age, penetrance could be really low because only 1 person has symptoms out of 10. And as you get older, up until the lifetime penetrance could be really high, 9 out of 10, for example. Again, this is not specific for CSF1R. I just made this up to explain what penetrance is, okay? So the key here is that penetrance increases over time. So we need to look at penetrance as a function of time, over time. Okay, so keep that in mind while we talk about the CSF1R protein, okay? So this is the protein right here. I want to focus on this part of the protein, okay? This is the part of the protein that's inside the cell and is the most critical part of the protein. It's called the tyrosine kinase domain, okay? And that corresponds to this part of the graph here in red with the little bars with other colors in here, okay? What I'm showing down here is a plot of all the 434 mutations that have been reported in CSF1R. And this is a diagram of the entire protein, okay? From the top to the bottom. This is the top to the bottom, okay? So it's already known that most of the mutations occur in the tyrosine kinase domain, and this is absolutely true here. But what I want to point out that's really interesting is that if you actually look at the tyrosine kinase domain, it's broken up into 3 major parts. There's an N lobe right here, okay, and there's a C lobe. Both of these lobes are really important for the function of this protein. And then there's a kinase insert domain that connects the N lobe with the C lobe. And this is a connector, so it's less important for the function of this protein, okay? If you look to see where the mutations, where the known disease-causing mutations are in the protein, they cluster with the N lobe and with the C lobe, and with the kinase insert domain, not so many mutations, okay? So the biology makes sense. What I also want to point out is that there are mutations outside of the tyrosine kinase. So there are mutations up here outside of the cell in the part of the protein that lives outside of the cell and binds ligand to activate this protein, okay? So I asked myself the question, can I, based on the knowledge, the known published knowledge of this protein, okay, the structure of this protein, can I then predict how severe disease is based on where it is, based on where the mutation is in the protein, okay? And again, this goes back to penetrance. Higher penetrance means more severe disease. Lower penetrance means less severe disease. That's another way to put it, okay? So I want to focus on these 3 parts of the tyrosine kinase domain, and I'm going to make an analogy to hopefully explain it in a clearer way, okay? So the analogy I'm going to make is to a circuit, like if you go into a room and you're going to turn on a light, okay? So the activation loop, this part, that is like the on-off switch of the circuit, okay? So you go into a room, you turn the light on by turning on the switch. And if the switch is broken, the light will never turn on. Okay, so we predict that that's going to be very high severity because the light will just never turn on if that's broken. The next part we're going to talk about is this, the catalytic loop, okay? So that directly alters the activity of this protein, and that's kind of like the light bulb, okay? So If you cut the wire inside the light bulb and the light bulb doesn't work, no matter how many times you turn on the switch, this light bulb is never going to come on, okay? But also, that's not the only way the light bulb can break. Sometimes, when you're screwing in the light bulb, you don't screw it in tightly enough and it is sort of connected, but not quite, so that when you turn the light on, it flickers. It's sometimes goes off, sometimes goes on. So that can also happen with the catalytic loop, okay? So we predict that this can be high or medium penetrance, okay? The last part I want to talk about is the alpha C helix. This part is like the battery or the electricity running through the current, okay? So for example, if it's a battery, when the battery is about to die, okay, the light gets dim, but it's still on, right? So it still works, it just gets dimmer, but it's still working, okay? So that's what happens in the protein when the alpha C helix has a mutation. The protein can still work, but it's not as efficient as when there's no mutation in the alpha C helix. So we predict that to be lowest severity. This is all relative to each other, okay? So I looked at the penetrance of these three parts of the tyrosine kinase domain, the most important part of that protein. Okay. Higher penetrance or a higher severity is going to be towards this side. Lower penetrance down here, okay? As predicted, the activation loop, which is the on-off switch, okay, has high penetrance. The catalytic loop, which has higher medium penetrance, has slightly lower penetrance here. To be totally honest, okay, there is no significant difference between those two lines, but there is a trend where the green line is a little bit shifted down and towards the right compared to the red line. If you look at the alpha C helix, okay, this is the one where we predict to have the lowest penetrance or the lowest severity. This line has lower penetrance, okay? So again, the biology and what we observe in patients correlates really well. So this is really important because for a very long time we've been saying for a lot of, not just CSF1R, but for a lot of these genetic disorders that there is no genotype-phenotype correlation. In other words, we can't really predict what your clinical presentation is going to be or how severe your disease is going to be based on your mutation, okay? So this is the first time where we can actually now start to see some patterns based on where the mutation is in the gene or in the protein. So this is really going to help us counsel patients, but I want to put a caveat out there, okay? Over time, the shading you see here, okay, so there's a line and then there's shading underneath the line. The shading is actually what we call the 95% confidence interval. So what that means is that there is still significant uncertainty, okay? When we talk to you about possibilities of X, Y, or Z or getting symptoms, these are all possibilities. We can say there's a higher possibility or a lower possibility, but we cannot give you an exact number for this exact reason. I do want to give you some numbers from this chart though, okay? So remember I said penetrance changes over time, okay? So I have age here, and penetrance will increase as age goes up. So for the most severe mutations in the activation loop, this is the on-off switch. By age 60, the penetrance is 96.8%. That means 96.8% of the patients who have mutations in the activation loop will develop symptoms by age 60, okay? Contrast that with the alpha C helix, which is the least severe area. 79.2% of patients develop symptoms by age 60, okay? And if you look at the change, the difference in penetrance between the most severe and the least severe, the biggest difference is actually here around age 50, and there's a 25% difference in penetrance. So for the activation loop, 87.1% of patients who have a mutation here will have symptoms by age 50, whereas only 62.5% here will have symptoms by age 50. So that is quite a significant difference. Okay, so without going on too much, because I know we're running behind, I hope I've shown you that the International CSF1R Variant Database is a powerful tool that we can use to understand this disease. And it's driven 100% by data from our patients. So I am very thankful to everybody here who contributed to this database. I also hope that I've shown you that this database can help us with variant interpretation. It's going to help us with counseling patients, okay, and giving you more accurate information. And what I didn't talk about here, but what is really relevant, especially in light of the other talks today, is that it's really going to help us determine the criteria for treatment, treatment timing, and also design of clinical trials. How can we stratify patients such that we can better measure outcomes based on where someone's variant or mutation is in the protein or some other quality of that particular mutation. But again, this is also the most important takeaway. You are not alone. There are a lot of patients with mutations in CSF1R, and I suspect that number will only grow as genetic testing becomes more available and people know about this disease and we increase awareness. And again, your story may be someone else's answer. So thank you for helping us out. Thank you to all our amazing collaborators. Without them, I would not have this database. And thank you especially to our entire community. I know I've asked many of you for data more than once, so I really appreciate your patience. I can take questions.
Oh, okay. We have a lot of questions, so can we make it quick? I know we're running behind, but is everybody feeling okay? We're okay if we go a little over? Okay.
So I have the variant, and a couple months ago, my dad, who's going to be 83, was tested positive. So I'm wondering, I'm assuming it's still valuable for him to be a part of the data. Okay. All right. That's my
Yes, thank you. Mm-hmm. Please get in touch.
So how much of your data are based on exome sequencing, and how would that change if you had all the data in the world on whole genome sequencing?
Great question. So I don't have an exact number for exome sequencing. I can tell you that most of the patients in literature were diagnosed either from leukodystrophy panel or exome. There were many fewer on whole genome, especially for adults, and that's due to many reasons, including insurance coverage. So exome versus genome certainly would make a huge difference. There's quite a few caveats about genetic testing. Well, let's start with this one. So when you send whole exome, okay, the name of the test is whole exome, but it doesn't actually give you the results for every single gene, okay? As a clinician, when you submit the test for whole exome, you have to write down what symptoms your patient has, and the genetic testing company, their genetic counselors will look at that list of symptoms and come up with a list of genes to look at. And there are many reasons for that, okay? And sometimes, okay, they may not find the gene because they haven't looked at that particular gene, okay? So that's one caveat in exome sequencing. That is also true for whole genome sequencing. However, where whole genome sequencing is much better than whole exome sequencing is identifying the intronic variants. So a huge proportion of the patients here actually have intronic variants. And so let me just explain to the patients here what intronic variants are. So remember that group of DNA like letters that makes up the gene, okay? That group of DNA of letters can also be divided into exons and introns. Exons are the parts of the DNA that make up the protein, but they are divided into small groups, and in between each group, you have other letters that don't encode the protein that are called introns, okay? So introns they don't encode protein, but a mutation inside an intron can also cause disease for reasons that I will not explain here, but that is a really important part of this disease because a lot of our patients have intronic mutations. So that would be where whole genome sequencing would be really, really helpful because there are a lot of deep intronic mutations. Yes.
You on the effort here because I know this is a huge undertaking, and I think that mapping the genome parts to the structure of the protein is something that the field is going to very much appreciate. So I want to congratulate you on that. A lot of hard effort. The next thing is I'm curious how often now we find a new mutation. So, you know, you're seeing these mutations. At what point, you know, are you still surprised when you find a new one, or do we have a pretty good understanding, do you think?
I am not surprised when we find a new one.
Okay.
So I will tell you the actual number of new mutations. I didn't include it on this slide because then I'd have to redo every single graph.
Sure.
The actual number of new mutations now is actually 436. I just got 2 new ones.
So 436 from when you started, or from what point?
From when I started.
Okay, so like a year and a half from now.
Actually about a year exactly. I started last year in May, a year and a month.
Can I ask a follow-up question on that,
Yes.
which is how much?
Go.
Okay. How much of this is, do you think, we're finding all the new mutations, what does that tell you about underdiagnosis?
There's tons of underdiagnosis, I'm sure.
Yeah.
And I can actually provide proof for that, okay? Yeah. And this is, I didn't present this data, okay? If you, so the penetrance calculated here, okay, suffers from ascertainment bias. What that means is that the penetrance calculated here is derived from patients who were published in the literature. So that means that the patients need to be published, right? So you are already enriching for the population of patients who go to a clinic, see a neurologist, see a geneticist, and then get published, okay? That means there are probably lots of patients out there who are not diagnosed or who are diagnosed with something else that have not been published that are not part of these calculations, okay? The paper published by my colleague, Dr. David Lynch, last year, I think it was last year, maybe the year before that. Thank you, yes. Looked at the UK Biobank, okay, and looked at CSF1R variants in the patients in the UK Biobank. And if you calculate penetrance if you calculate prevalence first based on Hardy-Weinberg equilibrium using the data published out of that paper, and then you calculate penetrance from that clinical prevalence data, okay, it is way lower than 90% or 80%, okay? So that tells me 2 things. 1, there's probably a ton of misdiagnosis and underdiagnosis. And 2, the true penetrance, okay, may not be as high as what is shown here. Now, I don't know how much of both of those things, how they're weighed, right? So which one is more, which one is less? And we need to do more studies to really understand that phenomenon. But I think those data are very strong evidence that there is underdiagnosis and misdiagnosis happening in our community. And frankly, I'm not surprised because of, you know, the barriers to genetic testing for adults. Okay, thank you.